I'm Mike Owen, and with the help of a number of people, Jim Lux, Damien Franzenberg, Ithy Cohen, McElvey, and Eric Jones a little bit, and Jim Lee, have organized the 29th annual Staniforth Memorial Lecture. We have gone outside the box a little bit, because while this is a lecture typically for weed science, if you look around the audience, what you will see is economists, and you'll see plant breeders, you'll see entomologists, and others. And not to put any due pressure on you, Fred, but you're sitting next to the press. They are from the Iowa State Daily. You will be featured in a story that is supposed to appear tomorrow in the Iowa State Daily, and I've also invited, if they have questions, to make sure we get it accurate, right? So it's an incredible honor for me. I have been working on getting Dr. Gould to be the Staniforth lecturer. I mean, I think he just did it because he got tired of me bugging him to do it, but last summer in Guadalajara, Mexico, he was at a meeting that I too was presenting, and I basically did a half Nelson on him and twisted his arm, and he agreed to come and present the 29th Staniforth lecture. Really, his credentials are incredible. He is a distinguished university professor. He is also the Reynolds Professor of Entomology. And Eric, you learned what Reynolds professors mean at NC State, correct? Just nod your head this way. So, he is also a member of the National Academy of Sciences. And so it is with great pleasure and tremendous honor from me to introduce our 29th Staniforth lecturer, who will present a lecture on will genetically engineered pests protect health, biodiversity, and crop production. Dr. Gould.
All right, well, it's really an honor for me to be here, and I've had a great day talking to a diversity of folks all across the board, from entomology to agronomy. And, when asked to come and give the Staniforth lecture in honor of a weed scientist in the agronomy group, I'm scratching my head and thinking, I used to be an entomologist, but this is interesting to be invited to do this. So I did want to show you that I had some credentials in this way, because in 1995, I published a paper in weed technology. So, so that was my claim to that. But actually, what I was doing was comparing strategies for managing resistance to insects and weeds because of their different biologies. And it was really an eye-opener for me to see how these different biologies might impact that. I'm not going to talk about that today, but I'll come back. I will show you at least 2 slides of weeds in this talk. So first, I just want to acknowledge the members of the lab who've worked on that. Alan Lloyd is my great colleague in the biomath, and most of these postdocs and graduate students have contributed to the modeling sections of all this. And then Tom Scott, Amy Morrison, and Elvio Aste were people who worked on the mosquito work that I'll be talking about, and we've been funded by the NSF and the National Institute of Health. So just to get started over there. So this is what I thought. This is a pretty general audience. I may actually, given what I've learned about the backgrounds, I may skip over some of the real detail, but I want to give you sort of concepts and history of genetic pest management. I want to tell you what it is, potential impact of these advances in molecular biology, and then the intersection of ecology, genetics, and public values in how this is coming forth. So first, to tell you about genetic pest management, what is, some people think about genetic pest management and they think of, oh, transgenic crops. No, we're talking about genetic manipulation of the pests themselves for controlling or manipulating that pest. So this is just an early example of a transgenic pest. This is Aedes aegypti, a typical Aedes aegypti under a fluorescent scope, and you can see a little autofluorescence here. but this is one that's been genetically engineered. And here you can see a marker gene in the eye that's fluorescing really brightly. And you also see fluorescence in the midgut. And this is a female mosquito about 8 to 12 hours after taking a blood meal. And this work was done by Alexander Franz, and the idea, what he was doing, was quite clever. Basically, what they did was they inserted a construct, a genetic sequence, that used a promoter from a peptidase gene, and a second sequence that created interfering RNA for the dengue virus. And the idea here is that if you want to slow down dengue virus, what you do is you interfere with one of the early genes that is necessary for the dengue virus to reproduce. But you could do that and just turn that on throughout the insect. But if you're doing that in the larval stage where the dengue virus isn't, you're just wasting energy and probably causing a fitness cost to the mosquito. So what they did is hooked it up to the promoter of a peptidase, right? The promoter is something that turns the gene on and off, right? And so it only was activated to make this transcript when there was a need in the mosquito to turn on production of a peptidase. And if you think about that, the adult male is mostly having nectar sources, not getting a lot of protein, but it's the female after she takes a blood meal that needs a peptidase, right, to break down that protein. So basically, this is pretty smart because when the mosquito takes in a blood meal, when it may also be taking in the dengue virus, that's when it gets turned on. So it gets turned on just when the dengue virus might be starting to replicate and shuts it down. So this mosquito was engineered with that, but to not transmit dengue 2 virus. It would transmit some of the others. But just as a quick slide, I'm not going to go through this in a lot of detail. This is what the construct looks like, right? So it has this carboxypeptidase promoter here. It has the coding for double-stranded RNA here, and then it has the reporter gene and so on to go in and be expressed. This is the only molecular biology slide I think I'll show you. Okay. So to go back to the general idea, right, there are two general approaches to using genetic pest management. One is to replace the strain, right, to have a strain that did transmit dengue, replace it with a strain that does not transmit dengue. The other one is suppression or local eradication. And that one is best understood through the early work of Edward Knipling in the 1950s. This is a slide when he was young, but he got the World Food Prize when he was a little older in 1992 for this. And just to give you the sense that this is not a new idea to do genetic pest management, the tools have changed. So his whole thing was sterile insect release technique. And in this whole talk, whenever you see a green insect, that's a natural insect. One that's red has been genetically manipulated, okay? So what his genetic manipulation was like a sledgehammer, right? To hit these male mosquitoes over the head, basically, with radiation and break chromosomes. And having those broken chromosomes, they still could mate and fertilize eggs. But as the embryo started to develop, the embryo would typically die because of the broken chromosomes and not having good genomic integrity. So when the male mated with the female, you'd have dead or sterile offspring. So the idea was if you could produce enough of these males and release them where they outnumbered the regular males, they would mate with the females, and if it's a 9-to-1 ratio, you'd get this 90% sterility or dead offspring. So if you had a population of 10,000 males in the local population, this is on a log scale, and you release 90,000 of these males, that's the 9-to-1 release, and if you kept doing that, then the population would decline to extinction. And if you look at this in terms of that ratio, you start out with a 9-to-1 ratio, 90,000 to 10,000 males, but that changes. If you look at this next slide, right, this was a 9-to-1 release, but as the population itself goes down and you're still releasing 90,000, it's even more effective, right? So it's inverse density dependence kind of in terms of the operation of this. So There were some initial successes. Some of you may know this stuff, but for the people who aren't the entomologists in the group, right? The success with the screwworm fly. The screwworm fly, this is its summer range originally. This is a little suspect to me, but this is the way it's mapped out. But in the 50s, they started the experiment where they were raising lots of these and releasing them on Sanibel Island, then in Florida. And they pushed them down by doing these releases repeatedly. They got extinction of the screwworm fly all the way down to Mexican border, and then they realized, well, they'd come back up, so they made an agreement with the Mexican government and slowly moved down all the way down to Panama. And the important piece is that the savings, at least from an economic analysis that was published once, is $1.5 billion per year. And the interesting piece to me is that it doesn't take that many flies to cause that much damage. So the numbers that you have to release are kind of small compared to if you're working with another insect like a whitefly, where there are that huge numbers in a single field. So they were doing that and were successful at that. And once they got it down to Panama, very recently, they built a new factory, the USDA and the Panamanian government, that can produce 60 to 120 million of these screwworm flies per week, right? And anybody who rears insects around here knows that that's a lot, right? So this is, I got to visit this facility. This is the inside of the facility. Think about how you mix up diet. This is how they mix the diet. I would come up to about maybe here. It's a huge factory, and this is the inside of the factory. These are rows and rows, trays of diet. You know, it doesn't smell very good in there because this is meat-based kind of thing. But screwworm, like a lot of insects, actually pupates in the soil, on top of the soil. So after it's done its job on the cattle, it would drop down to the soil. And here, after it's fed, it drops off those trays and winds up in these troughs here along the side. And then people come around and the larvae pupate and they collect a really interesting operation here. And then, of course, they're irradiated as pupae and then they're released out of airplanes to do their job at mating. The interesting thing is, with all that's been talked about releasing males, they can't differentiate males and females of these screwworm flies, so they irradiate both males and females, making it a lot less efficient, but indeed it did work. So the question is, you know, can we improve the efficiency and guard against environmental impacts? One of the things, this is done in the 50s and 60s, this is before the EPA existed, right? So the idea that you might have some kind of environmental effect of this was not on the books at all. And indeed, there's one study done before this was started that basically hypothesized that you would have an increase in deer and wild boar populations because they too were affected by the screwworm fly. And the anecdotal information indicates that that actually did happen. You know, if that was done today, we'd look into all of these kind of things. So that's the way of improving efficiency and guard against these environmental impacts. So the question is, can you go from releasing this huge number that could work with a screwworm fly where very low populations need to be suppressed to a more efficient approach where instead of 9 to 1, maybe you could release 1 to 9, right? Could you come up with some kind of tools that would enable you to make this 100 times more efficient or even more than that? And so I want to talk about that. And actually, this is Max Scott at NC State, who works with screwworm fly, and he developed transgenic screwworm flies. And what you'll see here is the NC State colors that he was very proud to come up with, the red and the white. But indeed, the white ones are males, and the red ones are females. And the females are overproducing a certain compound that will cause their mortality before they become adults, and he just harvests out males from this. Of course, what he's doing now is trying to have the females die before they become large larvae, so they don't eat a lot of diet, you know, before they do that. So this is already being tested, and he just got permission this spring from the Panamanian government to do field testing of this approach. And we'll see what the results are. I think he sees some fitness effects of this, but at least it's a lot better in the sense of just being able to release males. So I said that there were two approaches. So that was the suppression kind of approach. But I want to get to this strain replacement, which also is not a new concept. this concept of, replacing a strain, that transmits a disease with one that doesn't transmit a disease, or getting a pest species to start feeding on weeds instead of on the plant, all of these kind of things have been considered, and quite a while ago. So this was just the example of that. But the problem with this approach here, where you're changing an insect from transmitting disease to not, is if you release them into a population, it's not going to make a lot of difference if there are a lot of other insects in there that are transmitting disease. So if you release 10,000 mosquitoes that don't transmit disease, and you have hundreds of thousands that do, what have you accomplished? So what you need in this case is some means by which, when that mosquito gets released into the field, its genes will take over, so that this one mosquito, let's say, that wouldn't be able to transmit disease will then become the whole population. And that's a trick I'll talk about when we talk about gene drive. So, as I said, this was not something new. These folks were all involved in this. Vanderplank in the 1940s came up with a way of making these kind of hybrid tsetse flies, and actually did a successful experiment. Serebrovsky, who was in the Soviet Union during the Lysenko era when Mendelian genetics was outlawed, still did theoretical work on this. And actually, Chris Curtis rediscovered his stuff after he had reinvented the wheel, so to speak, because Serebrovsky's stuff had not, the literature hadn't gotten out into England at that time. So anyway, these folks did a lot of work on this. Chris Curtis worked for 15 years, mostly in India and other places, on trying these technologies out. There were other people who were involved. They were great population geneticists and understood the ecology of these things. But after 15 years, he wrote this paper, Genetic Control of Insect Pests, Growth Industry, or Lead Balloon. You got the idea of lead balloon doesn't float, right? And he decided it was basically a lead balloon because they had put so much effort into it and so frustrated by the fact that when they made these strains that they could use as replacement or gene drive, they weren't fit enough in the field to ever get anywhere. So it was interesting because that was just at the time that people were first starting to engineer Drosophila, right? So transgenics and insects were just starting. So this kind of question of they were about ready to give up, is molecular biology come to the rescue? And there's a little question mark at the end of that. So basically, molecular biology gave more genes, more precise genomic manipulation. Before they had very few tools I'll talk about, and more powerful gene drive mechanisms, which I'll again talk about as we go on. But the ecology is the same, and I'll get to that at the end of this talk, and we have more social concerns. So I want to talk about molecular biology within that context. So beforehand, when I was talking about suppression, all they had was the sterile insect technique. And in terms of strain replacement, we were working with translocations and these interspecific underdominances, which I'll get back to a little bit. But that was it. And now, what we have is there's a short list of the kinds of technologies that are available. So what I want to do in this talk is I'll pick out three examples maybe. The CRISPR homing as a means for eradication or suppression. And then I'll look at engineered underdominance and a little bit about Medea elements to just give you a flavor for some of these approaches and what they can do. So, start with a CRISPR homing. How many of you have heard of CRISPR-Cas9? Pretty much everybody. Well, not everybody, but a large number of you. Okay. So CRISPR was the molecule of, well, I can't remember, 1 year, it was the molecule of the year. So CRISPR-Cas9, I'll just give a quick review of it. It's an interesting system. The name comes from clustered, regularly interspaced short palindromic repeats, and I won't explain what that is because I couldn't. But it's basically hijacked from a bacterial immune system that was discovered. There could be a whole lecture on that probably, but the main piece was that basically it's composed of two pieces. One is Cas9, which is a nuclease, something that degrades nuclear molecules, DNA, and then a guide RNA. So basically these two components come together, And the guide RNA basically is RNA that has a specific sequence that recognizes a specific DNA sequence. So it basically carries this Cas9 to a specific site on the DNA, on the genome of the organism. And once it gets it there, the guide RNA doesn't have any action, but the Cas9, it's a nuclease, it's brought to that site, it does the cutting. So I always see it as sort of like the lame man and the blind man, right? Together, they can do this activity. So if you look at this in another diagram here, so here you see this big blob, which is the Cas9, and the guide RNA that has a part of its sequence is exactly the opposite of the DNA, and it binds here, and then you have that cutting. There's a lot of detail to it, but that's the basic kind of program. And this is what it looks like, in a more diagrammatic thing of the protein itself, the guide RNA, and the extra part of the molecule coming together, and you see the DNA through here and how it grabs onto it. So, to get to the real point here, this is sort of a natural immune process in the bacteria, hijacked and been able to be used actually with mammals and insects and other things to actually cut DNA. But the important thing to realize is it combines also with processes that we already have in humans and insects and such, and that is DNA repair mechanisms. So if you think about this, all of you are exposed to UV radiation and other things that causes breakage of DNA. So you've evolved with a means of repairing that DNA. And there are two just general kind of approaches for repairing DNA. One is called homology-directed repair, where when you get this breakage in one of the strands of DNA, remember you're diploid and you have another strand, that is used as a template to repair this. But what you can do is when you have that breakage, you can put in a large amount of sequence, right, that has ends to it that are homologous to where the break happened, right? So you have a whole sequence that mimics the spot where that break happened that you cut, right? You know exactly where you want to cut it. You know what the ends would be that on the other side of those cuts, and you design pieces that would just mimic that. So typically, you're just repairing the DNA with the other chromosome that hasn't had the breakage, but here you're mimicking that, so it sort of fools the repair mechanism to bring in whatever you have between these homology arms, and that's why it's called homology-directed repair. So what you wind up with is that chromosome that you tried to cut. You cut that with the CRISPR-Cas9. You can entertain putting in your favorite gene. All right, and that becomes part of the chromosome, and then that's inherited. Now, people are using that for everything from trying to do genetics on mice, to crops, to humans, right? But in terms of genetic pest management, this paper came out in March 2015, which was actually part of a thesis project of Valentino Gantz, a single PhD student, right, who was trying to work less. And you can see the title of this is a little bit enigmatic. The mutagenic chain reaction, a method for converting heterozygous to homozygous mutations. Does that sound like genetic pest management? Not exactly, but in interaction with other people, he realized that this was a means of gene drive, and that's why it wound up in science. So what this means is that somehow you're able to cause a mutation that turns an individual that was heterozygous for a certain trait into one that is homozygous. So let me show you what they were doing. So I told you that the idea would be with this homologous binding is that you would have a genome, right? This is homolog 1. Let's say you have one of your chromosomes, and it has a certain sequence, this red sequence, right? And here is the construct that you're trying to put in, and you have these homology arms, HA and H2, that are just like that sequence that's in red in the naturally occurring chromosome. But instead of putting any gene in the middle, what they were doing was actually putting the guide RNA and the Cas9 in the middle. So what happens here is that this construct produces the Cas9 and the guide RNA. They come together and they cut right here. Once that's cut, this homology-directed repair uses these homologous ends to figure out just exactly where it's going to be, and then it uses this as part of the repair template. So what you wind up with is the repair leads to insertion of Cas9 and guide RNA in that one homolog. Right? So instead of just any gene, you've put back the Cas9 and the guide RNA. So think about that. What you wound up with is an individual that's a heterozygote, right? You have one homolog where you've inserted this, and the other one doesn't have that. Right? But now, this Cas9 and guide RNA have promoters. It's not showing all the detail, right? They're going to, again, make the Cas9 and the guide RNA. That's going to, again, go and find that spot in the other homolog, right? It's going to cut that. Again, you're going to have this homology-directed repair. That Cas9 and guide RNA from the homolog 1 is going to insert in homolog 2, and poof, you've gone from being a heterozygote to being a homozygote. And that's this mutagenic chain reaction. And it's called chain reaction because you have that homozygote, and that homozygote, if it's in the field, would mate with another individual that doesn't have the gene at all. Their offspring would be heterozygotes, but then their heterozygotes would undergo the same process. So that's where that chain reaction would go on. And that chain reaction turns into the whole population, turning into ones that have that. So now, if you have that, and you also don't only put in this Cas9 guide RNA, you also put in your favorite gene, like one that doesn't allow the mosquito to transmit dengue, then you have a system. So just to tell you what they did, they were interested in doing this. What he was trying to do was mess with the yellow gene in Drosophila to basically, which is a recessive trait, so you can't see it when it's a heterozygote. So he wanted to make it quicker, and that's what he was doing. So they had this construct that put the Cas9 and the guide RNA and this, and they used that to interrupt the yellow gene, which in Drosophila is the opposite, right? When you get the mutation, instead of a regular colored Drosophila, you get one that looks yellow. And so what they got when they had, this is the typical wild type or heterozygote, and this is the homozygote for that yellow color, right? So remember, the heterozygote looks like a wild type. So what's interesting is they did an experiment where they did a cross where they expected half yellow and half regular, and this is what they got. Right? So you can see that this is what's called super Mendelian inheritance, not a 50-50, but increasing that. And that's that conversion of heterozygotes to homozygotes. So to just rehearse, if you have Mendelian inheritance and you are a heterozygote and you have this one mutation and you mate with somebody who doesn't have it, right, then you're going to have a wild type and half wild type and half will get your gene. And so all of these would be regular types because there's no homozygotes for that allele. If instead you have that homing, then this male, instead of producing half its sperm with the gene, all of them have it, and it mates with a wild type, and then you get all heterozygotes. You still don't see that the first generation, but then when the female and the male have that, then all of them turn into homozygotes, and they're all yellow. And if that yellow is not good for survival, then in the field, then they would all die. Right? So if you actually look at that in terms of a population genetic process, if you look, this says homogenenuclease, which does the same kind of thing. If you release them at about 10% of the population, over time they'll increase in frequency until they get up to about 90-95% of the population. And this is if it's causing mortality of those individuals. So if all the individuals that are homozygous die. And just to give you a sense of why that happens, When you release it at a low frequency, if you remember your Hardy-Weinberg equation, this is not the exact equation, but it gives you a sense of it, right? When you're at a frequency where the wild type is at 0.9, then you only have really 1% of the individuals become homozygous, right? It's 0.1 squared. And then you get PQs, 2PQ is the number of heterozygotes, and then you have the wild types. But the important thing here is that only 1% of the individuals are going to die off here, but all this 18% is going to convert to the next generation after that killing happens to becoming homozygotes for the next trial. So you've increased the gene frequency, and this starts going up and up and up. And then there becomes another end where when you're at a very high frequency, this is at 0.9, you're producing 81% homozygotes, 0.9 times 0.9, and then 18% heterozygotes, and then very few of the wild type. And what's going to happen is that whole 81% is going to now die. So that actually balances things off, because that mortality pushes the frequency down, and you push it up and you come to an equilibrium. All right, and that equilibrium point depends on how good the gene drive is working. So that gives you a sense of these things increasing, and under this kind of condition, you come to an equilibrium where only 19% of your offspring are surviving. Right, so that's pretty, useful for control. So the idea being that if you release that one individual with the gene drive, they can't transmit dengue or whatever it is, and they're in low abundance, but then they become in very high abundance. Perhaps not the entire population, but due to stochastic factors, and if you can make the gene drive good enough, you could replace that population completely. So that's the general concept of this gene drive. And this first paper was with Drosophila in March 2015. By December and January of the next winter, there were 2 papers, one here in Nature Biotechnology in Anopheles gambiae, the mosquito that transmits malaria. And this is using this gene drive to actually suppress female reproduction. So it wouldn't affect males, but only female reproduction. And this is moving into a target gene that is involved in reproduction to cause it not to function. And they pick genes that would be important in reproduction, but could function if one copy was present. So homozygote wouldn't reproduce, but a heterozygote would. So this is looking at suppression by this female sterility, in the sense that you start out with a heterozygote and there's a wild type, so the heterozygote becomes becomes homozygous, it mates with the wild type, and then most of the offspring are just heterozygotes. And it shows one here where it didn't work, right? But in the next generation, those would all become homozygotes, males and females, and the females would then die or not be able to reproduce. And again, it shows, it's not going to be perfect. So they picked 3 different genes involved in production of offspring in embryo development, and they picked ones where they could look at the wild type and look at number of larvae produced when they block that gene in heterozygous form. They got just as many larvae produced as with the wild type, but the homozygote didn't produce any offspring. And they found that same kind of pattern for all 3 genes. And they thought, oh, wow, we have that. So they made these constructs, and unfortunately, it didn't work quite that way. Instead of it being like the wild type, the first one only had 4.6% that produced, 0 and 9.3. So they threw out these and used this one. And basically what's happening is the promoter was not specific enough, so that in the soma of the insect it became homozygous, so that it actually wasn't reproducing. But they took this one, which was on the edge of being potential, and they did an experiment with it where they introduced it here at a frequency of 50% and then watched what happened over time. And if you see this black line is what the expectation was for increase in the frequency using a deterministic model, and these gray lines were a stochastic model of what might have happened in these populations, and the red lines are what happened in 2 replicates. So it looked like it was good. And they published that in Nature Biotechnology in 2015. I mean, just like 9 months after that first paper in Drosophila. But then they published another paper in 2017. And what you can see here, this is the part of the paper that happened in 2015, and they kept those things. So this is the line of what should have happened, and this is the line of what actually happened. Not good. Okay? So what was happening here is I told you all about homology-directed repair. You and Drosophila and Anopheles gambiae have 2 ways, at least, of repairing DNA. One is this homology-directed repair, and the other is non-homologous end joining. Non-homologous end joining for you and for the Drosophila and everybody is that you get this cut, and the way the repair happens is the 2 ends just come back together. Now, that's okay, that can happen, and then you're back functioning normally, except if you have this overabundance of the CRISPR-Cas9 in your system, if you cut it and you come back and you're fine, then the CRISPR-Cas9 comes back and cuts you again. And it keeps cutting until some kind of indel appears, right? Something gets left out or inserted or a mutation happens so that when it comes back together, the CRISPR-Cas9 can't find it anymore. And by the time that happens, you've often knocked out the target gene, right? Because you've changed it, but also it's become resistant to your CRISPR-Cas9. Right? So sometimes the gene actually does function because you've taken out 3 base pairs or something and just 1 amino acid. And under those conditions, you have an insect that's resistant to the gene drive. And that's basically what they found here, was that that happened in their production scheme. So it was working good until these other individuals sort of took over the population. So this was a problem. Well, another paper came out that same year, and this is in PNAS, on doing the same thing with Anopheles stevensii. This is another vector of malaria that's a major problem in India. And here they were doing this replacement where basically they were inserting a construct that would decrease the reproduction of the plasmodium. Again, not going into a bunch of detail, but they had this nice construct to put in and had it hooked up to CRISPR-Cas9 so it would continue to reproduce. And this they were putting into the white-eye gene, but that's not too important. Again, not getting into detail, what happened with this was it worked fine in males, but it didn't work in females. And the females were producing, when the female carried it, produced these kind of non-homologous end joining. And without going into too much detail, when a male had it and had become homozygous, if it mated with a wild-type female, this would be the sperm entering this would be the wild type. And once the embryo was formed, then you would be starting to produce in the germline this CRISPR-Cas9 because the males were not functional in the beginning, but then once you had the embryo development, you'd be doing that kind of, the CRISPR-Cas9 and the guide RNA would be produced, and that would lead to everybody becoming homozygous. So you wind up with an individual where the germline is homozygous, but not the soma. But this is what's transmitted, is that homozygosity. But when you look at it from the female side, and this is from Tony James's paper, you see that if the female is the one that carries it, even at the egg stage, when the male gets there, the Cas9, this blue color, or bluish-green color, is being produced, which is the Cas9, and CRISPR. And that's causing this to cut, but it has nothing to pair with. And so it has to undergo repair by the non-homologous end joining. And what you wind up is these resistant types as you move through. And that's what was being seen here. You'd have the drive from the males, but not from the females. So this is my slide here of gene drive, phase of innovation. You know, when people were working on this way back in the 70s and 80s and thinking about this and came up with transposon drives and other things, you know, there was a little excitement, but then it sort of died out. And then with the advent of homing endonucleases and CRISPR and all of that, people got really excited, and especially with that publication of that first paper with Drosophila. And then all of this resistance started showing up. And so the excitement has gone down, not this low, but the question is, what's going to happen? My sense is kind of optimistic that people are going to be able to come around with approaches, and I could talk about it in questioning, with ways of making this work so that you don't get as much of that non-homologous end joining. So on the other side is this kind of concern that these mad scientists are going to get something that works so well that once they let it out of the lab, it goes everywhere. And I use this analogy to Kurt Vonnegut's Ice 9, where you train, water to crystallize at a higher temperature, and it just spreads throughout the world, and everything becomes ice. This idea that once these scientists who aren't thinking beyond the laboratory release these things, that if you do this, you could get rid of an entire species, and what's the impact of that on the environment and so on. I'll get to that maybe later. But what it leads to is for people to be able to think about, could you have a gene drive that would be spatially limited or temporally limited? So you could release it, it would do its job in a different way. And I give you engineered underdominance as an example of this. Remember, I mentioned these folks here years ago, and they were thinking about underdominance. And this is just giving you an idea of what engineered underdominance is. So most of you know what dominance is, right? Where you look like one of the parents but not the other. And overdominance is when the hybrid, you get sort of a hybrid thing. Underdominance is where you have big A, and little a, and they may be a little bit different in fitness, but their offspring don't do well. So there's a population genetic approach to looking at this, and what you have here are 3 equilibria. If you release, this is just one idea with translocations, if you release them at about 50%, this translocation, or this, big A, right, you wind up with an unstable equilibrium just at that 50%, because if it goes a little above it, then the translocation gets fixed. If you go a little below it, the translocation is lost. Okay? So if you do that and you release it high enough, you can replace a regular insect chromosomal approach with a translocation. And that's what these folks years ago were working on. And this has been done with engineering to build these kind of things. And so to explain to you why this happens, remember, you have your wild type and you release a whole bunch of engineered types. And if you think about them, if a wild type mates with the engineered type or the translocated type, their offspring don't do well, right? So mating with the opposite type is a bad idea, right? But if we assume that all of these individuals mate with the closest individual near them that's the opposite sex, and we think about that, for the individuals you release that had the translocations or had this engineered underdominance, right? By chance, they're going to make 5 good choices of mates and 1 bad choice. All right, so mostly they'll be doing okay in terms of their offspring, but one of them's going to have offspring that won't be so good. But if you shift over and look at the wild type that's in the minority, they might make 5 bad choices to 1 good choice, right? And so their overall fitness in this population is frequency dependent, right? The higher your frequency, the more good choices you're making, the better you'll do. So if you go back to this picture, if they're both equally abundant, right, well, they're all making the same bad choices, but as one becomes even stochastically more abundant, it makes fewer bad choices, and so its frequency would go up, right? So that's how this would work, and if you think about that, if you have a trait like not being able to transmit malaria, you could increase that in the population. So there have actually been 2 papers on this, both with Drosophila, using this kind of approach to change populations. And these are all in Drosophila and kind of artificial in some sense, but at least proof of principle. So, how am I doing on time here? Okay? Yeah, I know, but I don't want to go over time. Do I have until like 5 after 5? Or? Okay. All right, we'll have some questions. But I'm going to actually skip this section. I just wanted to show you that these are general models, but there's ways of getting to much more detailed models, and that's what a lot of the stuff that we do is about. So you have this engineered underdominance And I showed you what happens, but it's much more detailed models. Look at the reproductive value of different individuals. Like, if you were going to actually do this, what kind of mosquitoes would you release? Would you release eggs? Would you release adults? And it's, without going into a huge amount of detail, if you release males and females and you release them at one age, you really need to release about 40%, but if you release them just when they're reproducing, of course you need to release fewer. But there are a lot of other things that affect what numbers you have to release. And actually, it's better in this thing to release males and females than just males. And you could do that if you have an insect that you're releasing that has the trait of not transmitting the disease. So there is a huge amount of detail in this. This slide just shows that if you're going to release the individuals, you're better off releasing 2 age classes than you are releasing the optimal age class. So there are a lot of counterintuitive results that come out of modeling, but they do lead to, if you're going to actually do these things, how would you do it? So, and there's also the issue of how do you release them? If you have an area, do you release them in the middle of the city, or do you release them in patches? And which of these is the best also depends on a lot of variables. And we do spend a lot of time modeling this stuff. And again, I don't want to go through a lot of detail here, but just to tell you that there are certain kinds of migration probabilities, how these things move, where it is better to release in a central area than in patches, and other points where it's better to release them as patches. And so you need to know a lot about the ecology of the organism you're working with. So to end this stuff on the modeling, which I'm not going to go into a lot of detail on, we do a lot of modeling for Aedes aegypti in Iquitos, Peru. And Iquitos, Peru is the largest city in the world that you cannot reach by roads. Okay, it's in the middle of the Amazon in Peru, and as you can tell by all of these tarps, it rains a lot, lots of mosquitoes. But it's a city of about 500,000 people. And so we built this model, it's called Skeeterbuster, that has all the details of Aedes aegypti in it. And I won't go through a whole bunch of things. It has stochasticity, it's spatially structured and stage structured. So it basically mimics all the biology of this one insect. And so we use this to figure out if you had Aedes aegypti with a gene drive, how would you release these things? And again, I just want to say that we need these kinds of models to look at the movement of the mosquitoes and other things. I'll be happy to ask questions, but I don't want to spend too much time on this. But just to say that, we can mimic what would happen in a release if each of these dots is a house in the city. How would you release these things? How would they move? And so on. And we're actually hopefully going to be starting on an NIH grant to actually test some of these hypotheses. So I'm going to leave this go. So I always get to this point of telling you that, the model is just helpful to help you think about these things. It gives you ideas of counterintuitive things that once you realize it, then you understand that you don't even need the model anymore. You get it, but you wouldn't get it without the model. So what I want to get to before I end is about this question I bring up in the beginning is, will these things help with health, biodiversity, and crop prediction? So what are the good and not so good targets? So I said ecology matters. So, you have this molecular biology to the rescue, but the same ecology. So, and one of the first papers on this outlined how you could use this CRISPR-Cas gene drive to help you with all of these possibilities, with aiding threatened species, controlling invasive pests for agriculture, controlling vector-borne diseases, and so on. And so I used this for just a touchstone, right, to look at these different things. So environment, what species could you use it for to help save endangered species? A great target, and people are working on this, are the Hawaiian honey creepers. Beautiful birds. They're sort of like Darwin's finches, started out from a very small population and expanded, except they're quite a bit more beautiful, I'd say, than Darwin's finches. Right? Look at these. I mean, incredible diversity of ecologies, but they've been on these islands that never had mosquitoes and never had bird malaria. And the birds on the mainland had evolved more resistance to bird malaria. Well, good old Culex came over to Hawaii and started transmitting bird malaria. And that's just one of the factors that's causing endangerment of these species. But the issue is that could you make this mosquito not transmit the disease or knock down its populations? And I just want to say people are working on that, looking for funding for this kind of an approach. But if you look at the frogs, which are the main thing that is pointed to, these would be a terrible thing from my perspective. Frogs are just amazing, right? You have all this diversity of frogs. But if you think about it, you'd have to go after, there's a chytrid fungus that is causing die-off of these frogs. So it would be great if you could have one genetic mutation to make all the frogs resistant, but they're a species, and you can't cross these boundaries. And think about these tropical frogs. Some of them are just incredible, but none of them have been reared in the laboratory. How are you going to get these transformations to occur? So I think we have to think, in terms of what are going to be our poster things for these, a little bit more about the ecology. So to think about pest management, I always think the cockroach would be a great thing if you could, people would be pretty into the idea of getting rid of cockroaches. But the thing about cockroaches is that they have a very well-guarded egg case. It's very hard with cuticle. And the way you inject eggs with the DNA that you want to put in there is with a very fine needle. And so that's not possible. But some people are working now on ways of injecting right into the ovaries. And that would really change things for this. So other insects like bed bugs that are very inbred, made in your homes, would be something, again, that would be great to get rid of, but you couldn't use this approach. I mean, think about releasing a whole bunch of bed bugs into an apartment. Doesn't sound like a great idea. And also, it probably wouldn't work because they're very closely related. So if we think about agriculture, one of the targets has been spotted winged Drosophila. And I don't know if you have that in Iowa, but it infests, it's an invasive that infests a lot of small fruits. And this, unlike the screwworm fly, the populations in one field could be as many as a screwworm fly in a county. So you'd have to release a lot. So the idea is could use a gene drive for this. And of course there's some concern, but it's an invasive pest and that's been considered. But if you look at the coffee berry borer, and it's a major pest and a lot of people are worried about it and are trying to do genomic studies on the coffee berry borer, they grow up as larvae inside a coffee berry, and it's a bunch of brothers and sisters, and they mate before they come out of the berry. So think about, let's not do this, right? You release a whole bunch in the field, but they're not going to be there. So this is where the ecology comes in, and of course, aphids that don't, have sex, not exactly prone to that. Okay, and western corn rootworm is actually something people are thinking about working with. And Marcel Lorenz at NC State has developed transgenic rootworms. I mean, this is, years from now, but potential when BT no longer works at all, right? But in agriculture, so what I want to get to is, I told you I'd have some plants in here, right? So, could you use this to suppress weeds? And the thing that happens here is like with that coffee berry borer, some weeds, if you look at this in terms of, this is a review paper on outcrossing rate, some weeds have 0 to 20% outcrossing, right? So they're very inbred. Some are completely inbred, right? And then there are others that are almost 100% outcrossing. And so, of course, if you released it and it was a selfing plant, you wouldn't get very far. So this shows Scotch broom that isn't very highly outcrossing. But to think about it, if you think about morning glories, right, there are different species with different amounts of inbreeding. But if they have even 50% outcrossing, there's a lot of genetic variability, and you release these gene drives and kill off all the ones that become homozygous, there's going to be selection for more and more inbreeding. And we don't know what selection for inbreeding will do in different things, but there are differences among populations of morning glories in the same species for the amount of inbreeding. So you probably select them to become resistant by being inbreeding. But interestingly, Palmer amaranth and water hemp that I've heard a lot about today are very interesting because the weed scientists, I think, know that these things have male and female plants. So they have to be outcrossing. So here's your weed that is resistant to glyphosate and other things. Would it be possible to think about getting rid of these using gene drive? And I don't think anybody's thought about it. I don't think there's anything like a transgenic farmer amaranth yet, but it's something to consider. I'm going to move through some of these quickly here. So I'll leave you with the last one I want to talk about is this whole thing about controlling vector-borne diseases. And this is the tsetse fly causes sleeping sickness in Africa, and Trypanosomiasis, right? So sleeping sickness for humans, but more damaging, causing mortality of cattle. And that impacts agriculture in Africa, right, in terms of being able to plow. This is the area where tsetse fly occurs, but it's also an area of incredibly high biodiversity on Earth. It's a real hotspot for biodiversity. So the question is, if you actually got rid of the tsetse fly, you'd allow more agricultural productivity in this area in Africa. And you could think of that as being this balance between humans coming out of poverty, maybe, and a loss of biodiversity. But it would be interesting if you had to deal with that. But one of the questions that comes up is, what would that development look like for humans? Would it lead to less people in poverty or potentially more people in poverty because of unequal distribution? So that's something that is very important. This is just a summary slide to say that this gene drive technology has this potential, but we lack some of the tools, and that it's critical to understand the details of the ecology, and it's important to understand the social context. And this just talks about the spatial drives that are limited and how those are harder to develop than the ones that would spread everywhere. So will there be a societal call for that? And I would just say that there's a nice National Academy of Sciences report on gene drives that includes both the technical as well as some of the social issues. And we have a group at NC State that started out doing this work on ecology, molecular biology, and population genetics to build these gene drive systems. But we realized, given the environment of this whole thing, we turned into a different model and put this as a major piece in what we're doing. So we brought faculty together across this whole thing, from molecular genetics to rhetoric and communication and ethics, and we formed a group, and that is our Genetic Engineering and Society Center. And we now have about 29 faculty across all of these disciplines trying to work on these issues, not just with gene drive, but with genetic engineering in general that brings these things up. So we were able to get a grant from the National Science Foundation to train students. This was a great grant of $3 million, and we've trained students across these disciplines with this. All right, well, I'll stop now, and if you have any questions. Thank you.
I'm sure that Dr. Gould will entertain questions. So, from the press.
I actually don't. So that beginning question that you started out with, what would you say the exact answer to that is?
I'd say there is no exact answer to that question. Right, so I think that it's still a question. I think that there are going to be, as I said at the end of the talk, there'll be specific systems where these could turn into really great tools. And jury is still out on how much we'll be able to get over that barrier of resistance evolving to them. So we don't know the answer to that yet. And that's why I say I'm optimistic that we'll get somewhere with it. I don't think that the concerns that people have that we're going to wipe out whole species, right, will become an issue, because we'll be lucky if we could wipe out a species within a county kind of area. But I do think that the technology is moving ahead quickly. So I would say that in terms of, my prediction would be, in terms of some pests, this will become an important tool. That's about as specific as I can get without getting myself in trouble. Yeah.
Is anybody working on this with the amaranths?
No, nobody is working on it that I know of. But actually, in that gene drive report by the National Academy, that's pointed out as one of the possible targets.
I did notice in your list of people, there aren't any scientists in that group.
Well, I have to tell you that we do now have some weed scientists who are interacting with us and coming to the meetings, yeah. Other questions? Yeah, do you have another question?
Yeah, that report, Gene Drive on the Horizon, where is that published?
It's the National Academy of Sciences, Engineering, and Medicine. If you just type gene drives on the horizon into a Google search, you'll find it. And it's free. You can download it for free.
If you could actually get to the point where you could eliminate a Drosophila species of tsetse fly, is that a bad thing? Would there be cascading ecological effects?
So those are the questions. Those are the questions that keep coming up. So it's interesting. So in terms of malaria, there's been a lot of effort in this area. In terms of especially Anopheles gambiae in Africa, and the question always comes up about if you got rid of Anopheles gambiae, would there be impacts? Are there any species that rely on Anopheles gambiae? For example, a bird that feeds on them, relies on them, or in terms of their ability as detritus feeders. And there is now funding from the Gates Foundation for researchers to study that in Africa to see what the impact is. But we had these meetings, right, and with African scientists and African public, and you ask that question of how big would the ecological impact have to be to counterbalance the morbidity and mortality caused by Anopheles gambiae and malaria. And just think about that. It's one thing to ask that question in the US, it's another thing to ask that to somebody who's had malaria, and we're worrying about one bird species or one insect species. So that's an interesting question, but where it's come more to the fore is in terms of conservation biology, right? So we are working with a group that's trying to eradicate mice from islands, right? Because mice are invasives on oceanic islands, cause a real loss of biodiversity. So there's a group called Island Conservation that has been dropping anticoagulants with helicopters over these islands and getting rid of mice and rats. And it's been successful in some cases, but it has had off-target effects. So the idea is, could you use a gene drive to get rid of those rats and mice on islands? And this is a case where we're trying to work on one of these where you have a gene that's fixed on the island, but not on the mainland, so it can't get to the mainland. But that's a hedgy thing, and there are people who are against using genetic engineering for anything for conservation. So it's been a big battle between the Union of Conservation researchers and people who are in favor of gene drives. There's a lot of that. And so those are detailed questions. The interesting thing I think of is if you go to New York City or Paris or Washington and you say you want to get rid of rats. They want to get rid of rats, and you tell them, oh, but if you got rid of rats, you might get rid of all the rats in the world. And what would most of the public say? But if you ask the researchers who are working on it, they'd see that as a big problem. So that's where these spatially restricted gene drives would probably be.
Well, we do have cases of,
Okay.
just a quick follow-up. We do have cases where an insect species has gone extinct, like the migratory locust, for example. And maybe we weren't around to see all the cascading effects, we weren't there to measure them. But that disappeared without a whimper, as far as we know, for other species.
Well, I just say that when we release any kind of biological control agent, like we released biological control agent in Africa, it's spread throughout Africa, we didn't even ask those questions of what that would do. So these are real questions, though. Somebody else had a question? Yeah.
You answered partially with the last one. I was going to ask about the spatially self-limiting gene drives. So islands would be an example, but could you give us a little bit more of a taste of what approaches or what kinds of examples that you might use to illustrate that?
So for spatially limited? Well, so the idea we have, for example, with the mice, okay, is nicely, mice are a good target because we know a lot about their genetics, right? But what we know from population genetics is when you go to an island, these populations were started with very few mice. So it's the same thing of when you look at human beings, if you look at Africa, you see more genomic diversity than you see in Europe or in the Americas because people leaving in smaller numbers decrease the amount of diversity. So on islands, you expect mice to have some of their genes fixed for a given allele, for a given sequence of DNA. But on the mainland, where you have more diversity, you'd have a broader array of those. So if you make your gene construct for doing the CRISPR-Cas9 work well on the allele that's fixed, the sequence of DNA that's fixed on the island, it could go straight through the island, eradicate them. If some of those mice got to the mainland, right, there would be some individuals that had that allele, but the ones that didn't have that allele would not be targets, and they would then continue to reproduce, and that would be lost. Does that give you an idea of spatial limitation? And there are other ones, of course, if you use that under dominance, that works, and I didn't explain that well enough, right? Remember, you had to get up to that 50% to get it to release. So you could release that in a city, it could work well, but then if a few individuals moved out, they'd be at low enough frequency that they couldn't increase. Thanks for bringing that up. Other questions? Yeah.
Just thinking about the point you made about the frequency of the individuals, the genes you're trying to drive into the population, they need to be above a certain threshold in order for that to work. And just wondering to what extent there's been thought around coupling those genes with a resistance gene, whether it's for an herbicide or an insecticide. The idea would be that it would basically knock down some of the population and increase the relative frequency of the genes that are trying to be driven into the population.
Let me see if I understand. You're saying that you do something to decrease the normal population?
Yeah, exactly.
Yeah.
By the gene of interest that's being released, it's trying to be driven into the population. Link that with the resistance gene to herbicide
Right.
or insecticide, depending on the species of interest.
So the idea would be that you're asking the question of, would it be feasible that the ones you're releasing would have like herbicide resistance and the wild population wouldn't, and that way they would increase in frequency, and then they'd have something else wrong with them later. That people have thought about that, but every time that has been brought up, it's been kiboshed because people don't want to see anybody releasing something that has resistance. No, but indeed, a lot of people, from who've done the population modeling, see this as a potential. But you can see why that doesn't go well with the public. It's like releasing bed bugs, right, in your apartment. Yeah. Was there another question?
Let's say